SwayamPrabha. Digital India. Educated India.
Till now we have mostly discussed about certain fundamental aspects of microfluidics, and of course with reference to some interesting applications, but in the last few lectures we will be mainly concentrating on some of the applications. Now when we discuss about applications of microfluidics, if you recall one of our introductory lectures we mentioned a lot of applications which focused on biological issues. So many times if somebody asks us that what are the applications of microfluidics, it is very tempting to say bio and non-bio applications. What it essentially indicates is that bio applications are very common and very significant. So bio applications in microfluidics are concerned are of concern from the viewpoint of several practical applications, and the entire paradigm of bio applications of microfluidics, that entire paradigm is often termed as biomicrofluidics. Now when we discuss about biomicrofluidics, we have to keep in mind that there are whole lots of issues of biomicrofluidics, and I do not have any intention of touching upon all the issues of biomicrofluidics in this particular course. Rather my objective will be to give you some glimpses of ideas relating to biological applications of microfluidics. So in the first part of the lecture we will be discussing about biomicrofluidics of cells and DNA. So in biomicrofluidics of cells and DNA we will be mainly concerned about the interfacing of microfluidics with cells or with DNA, or in general with cellular biology or with genomics for instance. So as we know that microfluidics deals with miniaturized devices and is mainly concerned with laminar flow. It requires lower reagent volume and reagent quantities. One can use it for high throughput screening. Microfluidic devices tend to consume less power, and one of the very big and important issues of concern why we intend to study cellular biology using microfluidics is that microfluidics can selectively address cellular length scales. Not only that, one can have precise volumetric and thermal control of samples, and you know in human body there is some natural microfluidics, biomicrofluidics. Like for example in human body you have a hierarchy of blood vessels, like you have large arteries, large veins, small arteries, small veins, arterioles, venules, and then microcapillaries. The microcapillaries are like microfluidic channels. So blood flow through microcapillaries is a natural biomicrofluidics taking place in human bodies. So as I mentioned that biomicrofluidics is an agglomeration of biology with microfluidics, and there are whole lots of applications that we talk about. Now as I mentioned in the first part of my lecture today, we will be concerning about cellular biomicrofluidics, and we will also give you some idea of multicellular biomicrofluidics, and but to begin with, like we have to clearly understand that why are we going for cellular microfluidics, what are the outstanding biological problems that we can solve using the the ideas of cellular microfluidics. Now there are several issues. Let me talk about one outstanding problem, say one particular problem in cancer biology, that is cancer metastasis. So you know that one of the most lethal stages of cancer comes into the picture when the cancer cell from its origin moves to a distant location within the same human body by flowing across the bloodstream. So when the cancer cell migrates in this process, it has to pass through the hierarchy of blood vessels that I talked about, and it also has to be passing through the microcapillaries, which are like microfluidic channels. Now the condition in the microfluidic channel is highly stressful, so that it may be very difficult for a normal cell to survive under such a stressful condition, but the cancer cell has excellent adaptability, so that it can get easily transported through the microcapillaries, and that is how metastasis can be sustained. So a big outstanding question is, what are the physical attributes of a cancer cell which gives them the capability of migrating successfully through the stressful condition of the microfluidic capillaries in human bodies. So understanding cancer progression is a very important and outstanding topic that can be discussed, or that can be I should not say that is yet satisfactorily resolved, but that can be probed to some extent successfully by using microfluidics. Now not only cancer biology, one has to understand that cells are typically of the length scales of let us say you talk about human bodies, typical biological cells are of length scales of around 10 microns or so. So the length scale of a cell is very much compatible to the length scale of a microfluidic system or a microfluidic channel. Therefore you can capture cellular length scales, you can capture the dynamics over cellular length scales in a microfluidic platform. Not only that, you can control the fluid flow in micrometers in precisely defined geometries of microfluidic cell culture that can mimic the in vitro cellular environment, and also can facilitate simultaneous manipulation and analysis starting from a single cell level to tissues cultured on fully integrated and automated chips. In diseased conditions, different properties of cells like morphology, membrane malleability, etc. significantly affected. So the alterations in the cellular properties affect the dynamics of cell in the physiological environment. So cellular dynamics in a microfluidic channel can give rise to the detection or indication of the possible existence of a disease. So because the conditions are different for diseased condition and normal condition. Not only that, microfluidics offers the possibility of delivering not only the chemical but also the mechanical signals, providing an extra degree of control. Now I can understand that this particular audience is not a biology specialist, neither me is a biology specialist, I am an engineer by training. So I think that it is appropriate that we touch upon some of the basic ideas or some of the basic concepts before we move on to our discussion, like for example, what is a cell? So the cell is the basic structural, functional and biological unit of all known living organisms. They are the smallest units of life that can replicate independently and are often called as the building blocks of life. Now cells within the human body contain thousands of genes, proteins and other chemicals enclosed within the cellular membranes, and each cell responds to the chemical signals from the body or the environment and modifies its behavior in response to the signals. Cellular diseases like sickle cell anemia, Alzheimer's disease and cancer occur when cells dysfunction. This may include the development of too many cells, deficiencies in existing cells, or dysfunction or loss of essential cells. So, as I discussed about the cells relating to abnormalities in the diseased condition. So, for example, you can see in this view graph the biconcave disc shape of a red blood cell. Now, this can be critically affected by genetic or acquired pathological conditions. Various diseases, I will not go into the details, will alter the morphology of the red blood cell, and when the morphology of the red blood cell is altered, if one can identify that properly, then that can give rise to the detection of several possible diseased conditions. Now, cells in channels, underlying motivation. So, importance of single cell analysis. Because of natural cell to cell variability in biochemical processes such as mRNA and protein expression levels, dynamical signaling processes and the inevitable stochastic molecular responses, population average bulk assays are often misleading compared to single cell analysis. So, one usually goes for cell culture platforms. I mean there are traditional cell culture platforms, but in microfluidics one can go for alternative cell culture platforms using microfluidics principles, and there are several advantages of microfluidics based cell culture platforms vis-a-vis conventional cell culture platforms. Why cell culture platforms are important? Because without cell culture it is not possible to study the behavior of cells. So, one of the basic premises of studying cellular biology is cell culture, and to culture cells in a microfluidic system the paradigm is significantly different as compared to the classical system. So, here we compare the conventional versus microfluidic cell culture system. In the conventional system there are large fluid volumes that prevent rapid changes. In a microfluidic system there are small volumes that allow dynamic control. Regarding addition of nutrients and removal of metabolites, in conventional system it is infrequent and manual exchange of large volumes. In a microfluidic system it is precisely measured, continuous or transient, but it is fully controlled. Stimulation with drugs, proteins and simultaneous imaging. This is a very important consideration, because many of the diseased conditions of cells or many physiological or biophysical conditions of the cells can be obtained by imaging. Now, in a conventional system it is mostly not feasible that you simultaneously give drugs and proteins and take the imaging, but in a microfluidic system it is feasible. Parallelization of cellular assays, in conventional system it is not feasible, in a microfluidic system it is feasible. Automation of cell culture tasks, in a conventional system it is possible, but bulky expensive fluid handling robots must be used. In a microfluidic system it is possible with a high capability of automation in compact inexpensive format, you do not need to use bulky robots. Single cell manipulation and analysis, in conventional system it is manually involved, inaccurate and low throughput system, and most importantly in microfluidic system you can do single cell manipulation and analysis very accurately with high throughput. So, there are several salient features of microfluidic cell culture: precise control of cell numbers and density in a given area or volume and placement of cells in complex geometries, accuracy and throughput of biological assays by orders of magnitude, spatiotemporal control over the concentration of biomolecules, allowing for instance the generation of diffusion based gradients of the given molecules, low consumption of costly reagents, automation of microfluidic cell culture systems allows culturing of cells for several weeks under precisely defined conditions without any manual intervention, and it provides the possibility to deliver not only chemical but also mechanical signals, providing an extra degree of control over the cultured cells. So, there are several steps involved in a microfluidic cell culture system, like basically you prepare microfluidic channels through soft lithography, which is very common in biomicrofluidics applications. We have discussed about this earlier. Then for cell culture you prepare the cells and then seed the cells inside the microfluidic channel, and then you do an analysis which is mostly microscopy based. Now many times you may not be satisfied with a simple microfluidic channel, but you can make a complex network of biomimetic microfluidic devices, and you can study the cellular behavior. The whole issue is that like earlier we have discussed one example where we are having a targeted drug delivery through a complex network of microfluidic channels, and the drug is targeted towards cancer cells. So, what it will do to the cancer cells basically is, first it will create a blockage of supply of oxygen to the cancer cells or nutrients to the cancer cells. So, it will cause ischemia to the cancer cells. Not only that, what it will do is it will supply the necessary drug or the necessary chemical to the cancer cells. So, in order to do so, basically the drug loaded molecule, the drug loaded beads, so to say, I have shown you an example video that how these beads are designed to pass through the microfluidic networks, and in this way you can create an artificial animal model. So, instead of making a whole lot of trials on a animal patient or a human patient and then waiting for the results of trials, you can design your drug treatment or drug based treatment or chemotherapy, say in a cancer treatment. You can design the treatment and a priori assess the success by making an artificial network of microfluidic channels and then targeting the drug loaded beads to the desired cells which are having cancerous traits. So, in this way one can to some extent replace the traditional animal based studies with a in vitro microfluidic system. Now, what are the outstanding issues in a cell culture based system? So, in a cell culture system, the micro system is important and interesting, the small scale system, because we have already discussed that it can resolve the dynamics over cellular length scales. So, in that to that extent the micro system is fine, but the question is, why do we require a fluid flow? Well, we may require a microfluidic conduit, but why do we require fluid flow? So, let us look into this. So, if there is no fluid flow in a cell culture system, then dissolved oxygen concentration increases and decreases and carbon dioxide concentration increases in the vicinity of the cell. So, this is because of lack of oxygen supply. Toxic metabolic products accumulate, pH osmotic balance is disrupted, leading to cell death. So, if you are trying to culture cells without giving rise to cell death, then obviously it is a matter of concern. So, why do you need to culture cells? Because you intend to study some biological aspects of the cell. Now, I mean, you may say that, well, we may not have explicit supply of oxygen, but the cancer cell or the normal cell whatever is being cultured, it is cultured in a microfluidic platform, and it let us say the platform has PDMS substrate, that is the microchannel is made of PDMS for example or PMMA. Now these are porous materials, right, these. So through the pores of the PDMS wall oxygen can be supplied. Well, to some extent yes, but it is not good enough to supply the necessary oxygen uptake rate. It is there, but it is not sufficient. So that means you must somehow supply nutrients to the cell by virtue of something else, and that something else is fluid flow. So that brings fluid mechanics into the picture in a cellular microfluidic environment. Not only that, you think of a biological scenario, you think of an outstanding problem in biology, that to understand fully the dynamics of a cell in a condition when there is incipient blood flow under a dynamic environment. Now if you want to mimic that in a in vitro system, that is in an artificial system, then in an in vitro system if you want to implement that, then what you require? You require a fluid flow past a cell. I mean the fluid ideally should be a blood analog, but even if it is not a blood analog, at least some like aqueous solution which should pass around the cell to mimic the biophysical environment in a real in vivo system. So we can understand the motivation of fluid flow in a cell culture based biomicrofluidic system. So one is a biophysical motivation, the motivation to represent the biophysical scenario in the human body, and the other is like to keep the cell alive you require to supply nutrients, and to supply nutrients fluid flow is one of the common means. Now here comes an interesting point. When you supply nutrients to a cell by virtue of fluid flow, then that nutrient will try to keep the cell alive, but the fluid flow itself creates a stress in the microfluidic environment and the cell may die because of stress. So on one side the fluid flow is trying to keep the cell alive by supplying the necessary nutrients, on the other side it is exerting stresses on a cell which if go beyond a critical limit can give rise to cell damage or even cell death. So the moral of the story is you require an optimal flow rate, not only that you require an optimal static incubation time before you start the fluid flow. Now one has to understand as a passing remark that when we are talking about cellular assays in a microfluidic system, one also needs to discuss a little bit about subcellular assay, that is measuring the chemical or physical function of property within a length scale that is less than typical cellular dimensions. That is also possible in the microfluidics based cell culture arrangement. So I can give you one or two examples, like intracellular transport from one side to the other has been studied by exposing different parts of the cell with different dye tagged macromolecules. If the size of the macromolecule is large, then diffusive mixing can be negligible. So you can study intracellular transport also, not just transport outside the cell, but what happens as a transport phenomenon within the cell. That is a matter of great interest, because within the cell there is an aqueous environment, there is a more solid type environment, there is a combination of aqueous and solid type environment, there is a transfer of mass and momentum within the cell. So the cell itself is a nice and interesting system to study, and that can be done. Another example I can talk about, you can in a reproductive system, when you have sperms, you can selectively isolate motile and non-motile sperms. Only motile sperms are able to move cross stream and non-motile ones are dragged with the laminar flow. Now we will move on to some issues of microfluidic cell culture based studies, and with certain preliminaries I will essentially give you some glimpse of some research that we have done in our group in this particular area, typically related to studying the adaptability of mammalian cells in physiological confinements to understand the mechanisms of cancer progression. So to begin with, like see, as I told you that in microfluidics you have a significant contribution coming from the engineering issues, and one has to understand that you are basically interfacing the engineering on the fluid mechanics with the biological scenario. And traditionally, I mean long times back, people from these two disciplines did not used to talk with each other very much, but with the advent of microfluidics and with the emerging growth of biophysics and biomechanics in general, it is now customary that I mean researchers from these two apparently or seemingly disparate disciplines, they combine together to solve outstanding problems in biology, and that is where the future of biological research is directing to. It is mostly interdisciplinary and not the traditional biological research that I mean some people perceive about. So let us say that you have a setup like this. So I will point out what are the relevant things. You have a pump, like for example a syringe pump or a peristaltic pump that drives the flow through the microfluidic system. The microfluidic channel has height h and width w. So you have let us say an adhering cell on a substrate. I will talk about the specific nature of the microfluidic wall, and you have a microscope based imaging system of the cell, and the image of the cell studied under certain conditions will give us indication of the stress distribution on the cell. Question is how that is possible, we will come into that. So it is possible through a particular type of technique called as traction force microscopy. So in the traction force microscopy, what you basically do is you embed some particles, these particles are embedded inside the microfluidic substrate. So these are fluorescent beads or fluorescent particles, and when the cell is sitting on a substrate and the cell gets deformed, these particles are displaced, and once these particles are displaced, you can take the image of these particles, and based on the image of these particles you can get by some mathematical analysis the stress distribution on the cell that could be eventually responsible for this displacement. So fluorescent level beads are embedded into an easily deformable substrate, and it is easier said than done because the substrate has to be significantly deformable for this method to work. Now not only that, the other point is that the displacement field of the bead embedded substrate is calculated by comparing the fluorescent images taken at identical microscopic location in presence and in absence of cells adhering to the substrate. Then images are compared by dividing both of these images in small viewports, each spanning over 10 pixel by 10 pixel, and locating the coordinates of the cross correlation function maximum through a 2D fast Fourier transform algorithm. So this entirely falls within the paradigm of image processing. So you can see that on one side you have microchannel, fluid flow, which is like a sort of an activity of a mechanical or a chemical engineer or a physicist, and on another side you have image processing, which is commonly dealt with by people from electrical sciences, and then based on these images we will be trying to solve some outstanding problems in biology, which is typically the work of a biologist. So how these activities are coordinated and how these activities are interfaced, that is a very interesting and nice aspect of not only interdisciplinary research in biology in general, but biomicrofluidics in particular. Now from the experimentally obtained displacement field, the traction force is determined by the unconstrained Fourier transform traction cytometry or FTTC method. Now as I told you that what are the unique features of this system. See, this traction force microscopy has been known for quite some time, but only a few years back, maybe around 5 years back, our group has been successful in implementing this in a microfluidic environment, which was not possible before. So how is it possible? You use a unique ultra soft substrate PDMS, ultra soft PDMS. So normal PDMS will have base is to cross linker around 10:1. Instead of that you make ultra soft PDMS. It is better, easier said than done, but it is quite difficult to make this kind of ultra soft PDMS with base is to cross linker ratio of 65:1. And then not only that, you make the substrate more cytocompatible, that is compatible to cells by modification of the PDMS substrate using APTMS and polydilysine. And then you have to integrate, not only you have to make this substrate, you have to integrate this with a microfluidic setup. Then smaller diameter of marker fluorescent beads like 0.046 plus minus 0.006 micron can be placed very close to each other. So typically 8 number of beads in 1 micron square area, allowing enhanced spatial resolution of the TFM. So in this way you can basically probe the dynamics of a cell over length scales of microns. So as I told you that what are the problems that you can study. So for example the traction force that is exerted on the cell as a function of time you can study with different fluid flow rates. So you can see that like after some time you can see that the media has to be replaced for the cell to survive. So optimum static incubation period is the period over which you do not supply fluid flow to the cellular environment. This increases with increasing volume per cell. So the traction force decreases after the optimum time. So after a critical time, if you do not supply fluid flow, if you do not supply nutrient, the cell is progressively going towards death, and you can program a cell death in this way. And so you stop the nutrient supply to the cell, that means you do not have any supply of nutrient to the cell. So after a critical time the traction force will decrease and the cell that signals that the cell is naturally going towards death, and in fact if the static incubation is controlled in this way, it can give rise to a controlled cell death or apoptosis. So like as I told you that there are several image processing issues involved here. So you can see that these are the these are the various facets of the experiment. So these are the fluorescent beads, and like this is the experimental setup. So you can see this is the microfluidic pump, and you can see the microscopes and the microfluidic setup here, and then you have a computer where you study the images. So the typical images, so you can see that you can see the contour of the cell adhering to the substrate. The red regions are the regions of high stress concentration and the blue regions are the regions of low stress concentration, and intermediate colors represent intermediate effects. So you can see that as a result of this study you can get a nice color map of distribution of stress on the cell. So with different flow rates you can see that the stress distribution on the cell is changing, and this is purely fluid mechanics. So fluid mechanics as applied to a deformable body. So if you have a cell, a big attribute to the cell that it is deformable. So in terms of fluid mechanics, it is the effect of fluid flow on a deformable substrate, but a substrate with not only deformability but a natural adaptability which is not possessed by an inanimate object. So as I tell in some of my lectures that in terms of fluid mechanics a cell is very similar to something like a vesicle, but the outstanding difference is that a cell has life whereas a vesicle has no life. So by virtue of the aspects of this living system, the cell will respond to the incipient fluid flow in a different manner as compared to what is done by a vesicle or any other deformable object. So you can relate the traction force and deformation, and so you can see the centroid of bead displacement can be plotted as a function of time, and this displacement is eventually correlated with the traction force that is distributed on the cell. Now so this is fine, but this is a purely engineering platform, but what kind of biological information we can extract from this engineering platform? Effect of micro confinement. Now what are the effects of micro confinement that we are interested to study in a cellular biomicrofluidic environment? One important aspect is tissue matrix confinement. So mechanical and chemical effects of interstitial flows that can be carefully studied. Shear stress near 1 Pascal is capable of affecting the cell physiology and proliferation. Not only that, there are chemical effects also. There are chemicals which are continuously secreted from cells. So there are certain chemicals, and we will study these very carefully. These are these may be autocrine morphogens, growth factors like EGF and all these. These molecules bind to cell surface receptors. So if you look into this cartoon, these are the receptors and the red dots are the molecules which are called as ligands which will bind to the receptors, and this can help the cell to assess the microenvironment. So cells are continuously releasing these chemicals or these molecules, and then these molecules are binding to the receptors located on the cell surface, and once this binding is there, then the cell can understand its microenvironment. But it is very important to understand that where does fluid flow come into the picture? The convective flux alters the spatial distribution of the growth factors, like for example the EGF or the epidermal growth factor. Now there are several other examples where the effect of micro confinement of the cell can be important, like blood flows through capillaries. Cancer cells preferably adhere and survive within small capillaries during dissemination. Size of blood vessels influences blood clotting dynamics, and mechanical signal transduction through EGFR is, or that is the epidermal growth factor receptor, is one of the critical issues by which mechanotransduction or transduction of mechanical signals in a biophysical system may be possible. So how do we study this problem? We make a simple schematic so that we can also study this problem from a mathematical perspective, that is we study the fluid flow and the species transport. So let us say that you have h as the microchannel height and h cell is a typical length scale of the microfluidic system. So the confinement factor is denoted by h by h cell. So that means if the smaller the value of h by h cell, that means that the stronger is the confinement. And in this particular cartoon, the epidermal growth factor is shown by the green colored circles and the corresponding receptors by the Y shaped symbols. So it is pretty clear that the EGFs are secreted from the cell and they are captured by the Y shaped ligands which are located on the cell surface. So as a problem of microfluidics, how do we theoretically solve it? We solve the species transport equation with a fluid flow which is typically a pressure driven fluid flow, and the boundary conditions are a reactive boundary condition at the substrate depending on the reactions that we are interested to study. So if in the absence of any reaction, the right hand side of the equation which involves the reaction kinetic features will be equal to 0. So although in many cases we are not interested to study reactions, I have purposefully put the reactive surface boundary condition to make the model look more generic. So then we study the EGF concentration as a function of the Peclet number. If you recall, the Peclet number is the ratio of the advection to diffusion strength or the diffusion time scale by advection time scale, and you can see that EGF concentration near the cell surface increases with increasing confinement. So confinement is a very important problem. See, in microfluidics there are two important issues that we need to address while modeling a real biophysical system in a human body. One is confinement, another is flexibility. The confinement is something, the confinement effect is something which we want to capture through the results that I am going to present here. The flexibility is a more critical problem because we can make microfluidic channels, with a good level of flexibility, but that flexibility may not essentially mimic the flexibility of a blood vessel in a human body. So as I tell you, and I often make this remark, that as teachers we always tell you that what are the problems either we as our own group or other research groups have been able to solve. Hardly we discuss in the class that what are the outstanding problems, and it is not a bad idea that we bring out an outstanding problem here. So if you want to make an artificial system which mimics the microcapillaries in human bodies, one aspect is confinement, which people have been able to successfully make, but the other aspect is the flexibility, and the flexibility real human body system, the flexibility of a capillary in a real human body system is very complex in nature, and that engineers have not yet been successful in manufacturing or fabricating microfluidic systems which mimic the flexibility of the blood vessels in human bodies. I mean there are several research reports which make flexible microchannels, and these are reported, well, those are flexible, those have controlled flexibility, but that does not essentially mimic the complicated nature of the distensibility of the blood vessel as a function of the local blood pressure as the blood is flowing through the capillary. So in these particular results we do not bring out the effect of flexibility, but we bring out the effect of confinement, and we have clearly shown that EGF concentration increases with increasing confinement. Then with this understanding, now what are the issues that we need to resolve with this little bit of background on creating a traction force microscopy based environment for studying the stress distribution. What are the questions that you would like to answer? Does the size of the surrounding confinement play a controlling role in dictating the effective survival capability of a biological cell? Where from this question is coming? We have seen that the EGF concentration increases with stronger confinement. That means the size of the confinement might play a controlling role in the biophysical response of a cell, and then it can give rise to a distinct survival capability of the cell depending upon the confinement. The second question is a more critical question. Well, the effect of confinement has to be there on a biological cell, but how is it different in cancer cells and normal cells? So, does the cancer cell possess better stress responsive ability within a micro confinement, or does it possess a worse stress responsive ability within a micro confinement? We need to not only get a qualitative answer to this question, but also a quantitative answer. That means that we know that confinement has its role on the survival ability of the cell. How is this role different between cancer cells and normal cells? So, this is a cartoon by which I want to demonstrate the whole idea. So, there are different types of cells. So, in this particular cartoon we have focused on adhering cells. Now when you look into the adhering cells, you need to carefully understand that as human beings we stand on a surface with the help of our legs. In a similar way, a cell sits on a substrate with the help of its own legs, which are called as focal adhesion points. Now focal adhesion points on the basal surface are these ones, and then what happens when there is a stress on the cell by the incipient blood flow, then these focal adhesion points are first disrupted. Just like if I am walking and somebody is kicking at my back, then first my legs will be destabilized and I would be mechanically destabilized by virtue of the destabilization of my feet. Similarly, the focal adhesion points will be disrupted. Now by a unique mechanism, this disturbance, this mechanical disturbance will be propagated through the cell in terms of propagation of biological signal, and then what will happen? The most distant part of the cell membrane, which is called as the apical cell membrane, will understand that there is a stress. So when the apical cell membrane understands the stress, then what it will do? To cut the story short, there are different types of entities in the cell membrane. In cell membrane there is a fluid type of entity which is called as lipid, and there is a solid type of entity which is called as lipid raft. Now with the stressful condition, two possible things can happen. One is the lipid raft can move from cell membrane to inside the cell just to escape from the stressful condition in the cell membrane, and otherwise if it is still residing on the cell membrane, then the lipid raft may break altogether. So I give an analogy like this. Let us say that a dacoit has attacked my house. Now if a dacoit has attacked my house, see, I am not a very brave person, so I will not be keen to face the dacoits with my own fighting ability, but I will initially try to hide myself in the bathroom instead of facing the dacoit. So the lipid rafts, instead of facing the stressful condition, they want to escape to go from cell membrane to inside the cell. Now in this condition, what will happen? The cell membrane which had lipid rafts now will get devoid of lipid rafts under the stress. So, the cell membrane will become more fluidic or more malleable because it has now less lipid rafts than lipids, and in this process because it is malleable it can adjust its shape in a microfluidic confinement to survive the stressful condition, and this is the short version of the long story that we tried to unveil through our experiments. So, I will try to go through the experimental results on shear adaptation of cancer cells in a microfluidic confinement and the confinement effect. So, what we initially wanted to study is that what is the mobile fraction of in the cell membrane. So, that means basically you can also talk it in terms of the diffusion coefficient. So, what you can do is that you can make a study which is called as FRAP, fluorescent recovery after photo bleaching. So, you tag the cell membrane with fluorescent dyes, and then what you do is that you photo bleach a part of the cell membrane, and then after photo bleaching the fluorescent dye molecules will diffuse from the surrounding condition because of the concentration gradient, and the faster is the fluorescent recovery after photo bleaching, that means higher is the diffusion coefficient and higher is the membrane fluidity. Why is membrane fluidity important? Because membrane fluidity essentially is related to the depletion of lipid raft from the cell membrane. So, there are two important phenomena which are taking place. One is the stress induced focal adhesion disassembly, that is the disruption of the focal adhesion points, that is the legs of the cell which are the connections between the legs of the cell and the microfluidic substrate, and lipid raft internalization. So, there are two events, one is focal adhesion disassembly, another is lipid raft internalization, and there is a time lag between these two. This time lag is the response time, this is the time that the cell needs to respond to its microenvironment. So, understanding or getting a feel of the microenvironment is focal adhesion disassembly, and responding to that is lipid raft internalization. So, shorter the response time, that means the cell is responding quickly to the stressful condition. So, we have studied the variation in the response time in the parametric space of H star, that is H by H cell and shear stress, and we have seen from this map you can understand that the response time decreases if the shear stress is increased beyond a critical value, or so the first is the response time decreases if the shear stress is increased beyond a critical value, or when the H star falls below a critical limit, that is H by H cell falls below 70 micron and the stress is greater than 10 dynes per centimeter square. So, this is called as confinement effect. The confinement and the stressful condition combined together can reduce the response time of the cell for adjusting it to its microenvironment. Now, the second question that we addressed, that we tried to address, the confinement effect is more exclusive for cancer cells. So, we have studied several cancer cell lines and normal cell lines, and our study has revealed that cancer cells are essentially more malleable than normal cells. So, under a stressful condition the lipid raft dynamics is more prolific in cancer cells, the lipid raft internalization is more prolific in cancer cells, that makes the cancer cell possibly better shape adaptive or shape adjusting or malleable to survive the stress adaptive capability of to survive in a stressful condition in a microfluidic confinement. So, the cancer cells adapt themselves better, that is how they survive in a metastatic condition, and if we can understand the biophysics of this mechanism of cancer cell adaptation in a microfluidic confinement, then this can give rise to a critical understanding of the metastasis of cancer cells from a pure biophysical viewpoint. Now molecular origin of confinement effect. So we have discussed about the EGF and the EGFR, and we will try to understand that how the EGFR activated EGFR concentration is related to the confinement effect, and to relate it to the molecular origin we have plotted a ratio eta which is activated EGFR by EGFR, and we have found that a decrease in the response time is equivalent to an increase in the activated EGFR. So that means that in the confinement in a highly stressful condition EGFR is more prolifically activated. So that is that is one thing. Not only that, beyond the critical stress the EGFRs form clusters. So you can see in these view graphs clusters of EGFR that we have picturized. So now the EGFR can be activated in two ways. One is ligand mediated, another is ligand independent and flow induced. The ligand is the EGF. So it is basically called as a ligand receptor model. Ligand is the EGF, and what binds with the ligand the ligand itself binds with its receptor, that is the EGF receptor or EGFR. So you can use some anti-EGF which blocks only the ligand dependent activation, and you can use a chemical which can block both ligand dependent and ligand independent and flow induced activation. Now a very interesting point that we have observed is that below a critical shear stress the activation is purely ligand dependent, fluid flow does not play a big role. Beyond the critical shear stress the activation is predominantly ligand independent and primarily fluid flow induced. So that is where you can see that the molecular biology of the cell or the molecular behavior of the cell is influenced by fluid flow. So this is a very interesting point where fluid mechanics is interfacing with molecular biology. So you can see that like normally the EGF distribution or EGFR activation that should not be ideally dependent on the fluid mechanics, but by the traditional thoughts those are dependent on the biophysical or the biochemical features within the cell. Now we have shown that how these can be tuned by altering the stress condition as well as the confinement effect. So with the confinement effect you have both the ligand dependent and ligand independent activation. So you have the ligand dependent activation which is dependent on the EGF concentration, and ligand independent activation of EGFR which is dependent on the fluid flow. So EGFR activation depends on EGF which is ligand dependent and also on fluid flow which depends on the confinement effect as well as the stress due to the fluid flow. So just to conclude on our discussions on cellular dynamics, I want to highlight on a few other applications, not just the model for understanding the biophysics of cancer progression, like for example how can we use this for medical diagnostics. I can give an example of cell sorter. So you can see here, I will explain this figure, the cell solution is focused into a smaller confinement so that the whole fluid comes under a beam of laser, okay, and only one cell is included in a single cross section. So why is it important is because very narrow channel fabrication is costly. So you can instead focus the cellular solution to pass through a very narrow passage, not a artificial channel but a focused narrow area by which it will pass. Then a laser beam of specified wavelength illuminates the fluid. if the cell passing through the illuminated volume emits a fluorescent light, a voltage operated valve is activated and the fluid is further switched towards a particular location where the cells will be collected finally. So, you can sort specific targeted cells from a cellular solution by using this. So, for example, in a blood sample of a specific volume, the number of leukemia cells can be determined in this process. So, cellular biomicrofluidics you can also use for medical diagnostic applications. So, we have discussed about some aspects of cellular biomicrofluidics, and in the next lecture we will be mainly focusing on microfluidics with DNA and some aspects of microfluidics based medical diagnostics, which is one of the very important areas of biomicrofluidics. Thank you very much.